Good day, students. You are welcome to our lectures for today. And today we'll be we'll be discussing about beta oxidation of saturated fatty acid, which we said that it has 4 enzyme catalyzed reaction. The first reaction is dehydrogenation of the fatty acyl-CoA that produces a double bond between alpha and beta carbons, that's carbon 2 and 3, which forms trans delta 2 enoyl-CoA. So you remember palmitic acid was activated to be able to transport it to the mitochondrial matrix for oxidation as palmitoyl-CoA. Now, it undergoes what we call dehydrogenation, and this is catalyzed this reaction. In the first step, we have acyl-CoA dehydrogenase, which requires FAD to give us FADH2. Now, this is our now product that is formed as trans delta 2 enoyl-CoA, and this acyl-CoA dehydrogenases, they actually have the reaction is catalyzed by 3 isozymes and of this acyl-CoA dehydrogenase, and each of them have their own specific specificity for the chain length of the fatty acid. Very long chain acyl-CoA dehydrogenase is acting on fatty acid that has 12 to 18 carbon, and we have medium chain acyl-CoA dehydrogenase MCAD that acts on fatty acid 4 to 14 carbon, and we have such short chain acyl-CoA dehydrogenase SCAD, which act on 4 to 8 carbon atom. And all these enzymes, they are flavoprotein that acts that has FAD as their prosthetic group, and the FADH2 that is actually produced is reoxidized by transfer of 2 electrons to an electron transfer flavoprotein ETF in the mitochondrial matrix, which in turn passes the electrons to coenzyme Q of the respiratory chain and with the concomitant production of energy. Now in the step 2, we have the second reaction in the beta oxidation cycle, which now water is added to double bond of the trans delta 2 enoyl-CoA to form the L stereoisomer of beta hydroxyacyl-CoA, or we call it 3-hydroxyacyl-CoA. And this reaction is catalyzed by enoyl-CoA hydratase, which is the reaction is analogous to fumarase reaction in the citric acid cycle. Now, in the third step, we have L-beta hydroxyacyl-CoA is dehydrogenated to form beta-ketoacyl-CoA by the action of the beta hydroxyacyl-CoA dehydrogenase. And this enzyme requires NAD as the electron acceptor. So we have L-beta hydroxyacyl-CoA, which is now catalyzed by beta hydroxyacyl-CoA dehydrogenase using NAD plus as our electron acceptor to give us NADH plus H, which we have now beta-ketoacyl-CoA. Now, this enzyme is absolutely specific for the L stereoisomer of the hydroxyacyl-CoA, and NADH that is formed in the reaction donates its electron to the NADH dehydrogenase, which is an electron carrier of the respiratory chain, and ATP is formed from ADP as the electrons pass to oxygen. Now, the reaction catalyzed by beta-hydroxyacyl-CoA dehydrogenase is closely similar or analogous to malate dehydrogenase reaction of the citric acid cycle. So if you remember citric acid cycle, that will help you to actually understand better what we are discussing about. Now in this last step, which is step 4, is actually a thermolysis where we have acyl-CoA acyl transferase, which is called thiolase. It now promotes the reaction of beta-ketoacyl-CoA with a molecule of free coenzyme A to split off the carboxyl terminal 2 carbon fragment of the original fatty acid as acetyl-CoA. Now this reaction now introduces a CoA, and we have this enzyme thiolase now splitting the carbonyl 2 carbon fragments to give us what we call the acetyl-CoA. And this acetyl-CoA is produced from, this is one cycle of the beta oxidation. We have one acetyl-CoA that is produced, and we have the fatty acyl-CoA that we started, palmitic acid or palmitoyl-CoA is now shortened by 2 carbon. We have acyl-CoA now as our the 14 carbon activated myristic acid, which is in form of what? Myristoyl-CoA. Now, the other product is the coenzyme A ester of the fatty acid, now shortened by sucarbon. We have explained that. And this reaction is called thaiolysis, and by analogy with the process of hydrolysis, because beta-keto-acyl-CoA is cleaved by reaction with the thiol group of the what? Coenzyme A. Now, the 4 beta oxidation steps are repeated to yield a molecule of acetyl-CoA, right? And we have a molecule of acetyl-CoA, right? So, and we have ATP that is actually produced. So, and this now is overall total, we have 8 molecules of acetyl-CoA that is produced as a result of what? Beta oxidation of palmitic acid. And the cycle is in 7 steps, and the 16 carbon now give us what? 8 molecules of acetyl-CoA. The 7th cycle, because the 7th cycle will now entail a 4 carbon acyl-CoA, right? Now, when that splits off into 2, we have 2 molecules of acetyl-CoA, making a total of what? 8 molecules of acetyl-CoA. And the acetyl-CoA produced from the oxidation of fatty acid can be oxidized to carbon dioxide and water by the citric acid cycle. And this table summarizes the total energy, total ATP that is produced as a result of oxidation of palmitate-CoA to carbon dioxide and water, and the number of NADH and FADH formed, and the number of ATP that will ultimately be formed when you have mitochondrial oxidative phosphorylation producing 1.5 ATP for FADH2 and 2.5 ATP for NADH oxidation. Now, for acyl-CoA dehydrogenase, we have 7 FADH2 that was produced, and if you calculate, we have 10.5 ATP. Beta hydroxyacyl-CoA dehydrogenase 7 NADH, that's 17.5, and isostrate dehydrogenase in the TCA cycle produces, you remember, acetyl-CoA will also enter TCA cycle for complete oxidation. So now produce 8 NADH is good to remember TCA cycle. So 8 NADH will give us 20 ATP, and alpha ketoglutarate dehydrogenase also give us 8 NADH. We have 20 ATP. Succinyl-CoA synthetase, which will now give us 8 ATP, because GTP is produced directly in the steps to yield ATP in the reaction catalyzed by nucleoside diphosphate kinase. So succinate dehydrogenase also give us 8 FADH2, which does 12, and we have malate dehydrogenase reaction will now give us 8 NADH, which is now 20. Now overall total, when we oxidize palmitate-CoA to carbon dioxide and water, we have 108 ATP. So you can, as an exercise, you can now calculate the yield of ATP during oxidation of 1 molecule of the following saturated fatty acid to carbon dioxide and water: arachidonic acid, 20 carbon, and lauric acid. This is an exercise. It's very important that you try to do these 2 assignments. Now, we have what we call hibernating animals, and fat bears, we call them as fat bears carry out beta oxidation in their sleep. That is a grizzly hibernating bears. So many animals depend on fat stores for energy during hibernation and during migratory periods and in other situations. One of the most pronounced adjustment of fat metabolism occurs in hibernating grizzly bears, and these animals remain in a continuous state of dormancy for periods as long as 7 months. Unlike most hibernating species, the bear maintains a body temperature of between 32 to 35 degrees Celsius, close to the normal level, although expending about 25,000 kilojoule per day, which is almost 6,000 kilocalorie per day. And the bear does not eat, no drink, no urinate or defecate for months at a time. Now, experimental studies have shown that hibernating grizzly bears use body fat as their sole fuel, and fat oxidation yields sufficient energy for maintenance of body temperature, active synthesis of amino acids and proteins, and other energy requiring activities such as membrane transport. So fat oxidation also releases large amount of water as described and in our equations and in our beta oxidation and citric acid cycle, which replenish water lost in breathing, And glycerol release by degradation of triacylglycerol is converted into blood glucose by gluconeogenesis, and urea formed during breakdown of amino acid is reabsorbed in the kidney and recycled. The amino acid groups reused to make new amino acid for maintaining body proteins. So bear stores an enormous amount of body fat in preparation for their long sleep, and adults grizzly consume about 38,000 kilojoules per day during the late spring and summer, but as winter approaches, it feeds 20 hours a day, consuming up to 84,000 kilojoules per day. And during preparation for winter, bears can gain approximately 180 kilograms during a period of hyperphagia before going into what we call the hibernation. Now, this change in feeding is a response to seasonal change in hormone secretion, and large amount of triacylglycerol are formed from the huge intake of carbohydrates during the fattening up period. So now let's look at oxidation of fatty acid, unsaturated fatty acid, which requires two additional enzymes or reaction. So most of the fatty acids that are in triacylglycerol and phospholipid of animals and plants are unsaturated, having one or more double bonds. So these bonds are in the cis configuration and cannot be acted upon by Propionyl-CoA hydratase because they are in cis configuration. It has to be in trans before Propionyl-CoA act on them. So the enzyme catalyzing the addition of water to the trans double bond of the delta 2 Propionyl-CoA generated during beta oxidation. Two auxiliary enzymes are needed for oxidation of the common unsaturated fatty acid, an isomerase and a reductase. So example, when you have a mono unsaturated fatty acid, oilate, is an 18 carbon monounsaturated fatty acid with cis double bond between carbon 9 and 12. Is denoted as delta 9. And the first step of the reaction, we have oilate is converted to oleoyl-CoA, and like the other saturated fatty acid enters into mitochondrial matrix via the carnitine shuttle. You remember the activation of fatty acid and the carnitine shuttle. So now we have oleoyl-CoA then undergoes 3 passes into the beta oxidation to give us 3 molecules of acetyl-CoA. Now that will generate what we call cis delta 3 double bond in decynyl-CoA. So we have at carbon 3 we have a double bond but in cis configuration. So we have this enzyme delta 3 delta 2 enyl-CoA isomerase will now convert that to trans delta 2 do-decynyl-CoA, right? Now it's converted to trans configuration. Now we have beta oxidation of that will continue in 5 cycles to form 6 molecules of acetyl-CoA. So now since we have now the explanation, we have cis delta 3 enyl-CoA that was formed in the 3rd round, and it's not a substrate for acyl-CoA dehydrogenase. It's not a substrate for our enzyme hydratase, right? So the presence of double bond between carbon 3 and 4 prevent the formation of another double bond between carbon 2 and 3, and this impasse is resolved by a new reaction that shifted the position and configuration of the cis delta 3 double bonds, which we have already explained in the reaction. So we have example, polyunsaturated fatty acid with multiple points of unsaturation can require energy to get them through beta oxidation. So another problem arises with the oxidation of polyunsaturated fatty acid. When we consider linoleic acid, linolate, 18 carbon polyunsaturated fatty acid with cis delta 9 and cis delta 12 double bonds. So the cis delta 3 double bond formed after the 3 rounds of the beta oxidation is converted into trans delta 2 bond by the aforementioned isomerase. So we have our linoleal-CoA entering beta oxidation 3 cycle to produce 3 acetyl-CoA, but we have cis delta 3 and cis delta 6 double bonds. So we have our delta 3 delta 2 enyl-CoA isomerase now converting converting the cis delta 3 to trans delta 2, but we still have cis delta 3 at delta 6 at position 6. We have another double bond in cis configuration. So now when that occurs, and the acyl-CoA produced by another round of beta oxidation contains cis delta 4. Now in this case, we have cis delta 4. After that, cis delta 2 now undergoes and produces acetyl-CoA. Now we have trans delta 2 and cis delta 4. Now the dehydration of this species by acetyl-CoA dehydrogenase yield a 2,4-dienoyl intermediate, which is not a substrate for the next enzyme in the beta oxidation pathway. So what will happen? This is now circumvented by 2,4-dienoyl-CoA reductase, which is an enzyme that uses NADPH to reduce the 2,4-dienoyl intermediate to trans delta 3 enoyl CoA. Now, cis delta 3 enoyl CoA isomerase now then converts trans delta 3 enoyl CoA into the trans delta 2 form, and which is now customary intermediate in the beta oxidation pathway. Now, complete oxidation of the odd number of fatty acid we have, although we know that most naturally occurring lipids, they contain fatty acid with even number of carbons. Fatty acid with odd number of carbons are common in lipids of many plants, submarine organisms, and bacteria. So what we have seen before is oxidation of saturated fatty acid with even number and unsaturated fatty acid with even number. Now there is another fatty acid that have odd number chain, okay? Now, and they are also common. And how does the body now oxidize them? So cattle and other ruminant animals, they form large amount of the 3-carbon propionate, which is now propionic acid, 3-carbon, and during fermentation of carbohydrates in their rumen, right? So the propionate is absorbed into the blood and oxidized by the liver and other tissues. And small quantities of propionate are added as a mold inhibitor to some bread and cereal stores entering the human diet. So you see, it enters into the human diet. So now long chain odd number fatty acid are oxidized in the same way as the even number acid beginning at the carboxyl end of the chain. However, the substrate for the last pass through the beta oxidation sequence is a fatty acyl-CoA with 5-carbon fatty acid. At the last step, you have 5 carbon because it's odd chain now. Everything will occur except at the last beta oxidation cycle, which you have now 5-carbon acyl-CoA. So now what will happen? You have now when this is oxidized and cleaved, you produces what? Acetyl-CoA and 3-carbon propionyl-CoA. And the acetyl-CoA can be oxidized in the citric acid cycle. Of course, we know that, but propionyl-CoA enters a different pathway involving 3 additional enzymes. So in step 1, we have propionyl-CoA is converted into form what we call the D-stereoisomer of what? Methyl-manonyl-CoA. And this reaction is catalyzed by propionyl-CoA carboxylase, which has a biotin as cofactor. It uses ATP and to oxidize it to hydrolyze ATP to ADP and inorganic phosphate to form the D-methyl-manonyl-CoA. That's the first step. Now, in this enzymatic reaction, as in the pyruvate carboxylate reaction, carbon dioxide or its hydrated ion, bicarbonate ion, okay, is activated by attachment to biotin before its transfer to the substrate. In this case, propionate moiety. Formation of the carboxy biotin intermediate requires energy, which is provided by the cleavage of ATP to ADP and inorganic phosphate. So in step 2, we have D-methyl-manonyl-CoA undergoing an epimerization to form what we call L-methyl-manonyl-CoA. So it's epimerized to the L-stereoisomer, and this reaction is catalyzed by methyl-manonyl-CoA epimerase. And the last step, we have L-methyl-manonyl-CoA then undergoes an intramolecular rearrangement to form succinyl-CoA, which can then enter a citric acid cycle. Succinyl-CoA is an intermediate in the TCA cycle. Now, this rearrangement is catalyzed by methyl-manonyl-CoA mutase, and it requires as its cofactor 5-deoxyadenosyl cobalamin, or we call it what? Coenzyme B12, which is derived from vitamin B12. So this is the reaction. So now, this enzyme depend this adenosylcobalamin dependent enzyme catalyze intramolecular rearrangement in which a hydrogen atom and a substituent on an adjacent carbon atom exchanges places. So in the reaction catalyzed by the methylmalonyl-CoA mutase, the coenzyme A group migrates from carbon 2 to we have the carbonyl group that is attached to coenzyme A. Okay, now it migrates to carbon 2 and 3 in an exchange for hydrogen atom. So, okay, I think we can stop here so that when we come next, we'll discuss about fatty acid oxidation. How is it regulated? Okay, so thank you very much for listening and watching. See you next.